The point is that we would like to model the complex genetic disease acute lymphoblastic leukemia in childhood on an ex vivo platform where we look at drug responses of these primary samples. We are using a co-culture system where we culture primary leukemia cells on top of mesenchymal stroma cells that have been derived from the bone marrow of healthy persons. We grow the mesenchymal stroma cells as a first layer, and then we add the leukemia cells on top of them. We have learned first that the leukemia cells, they profit from this support. So we see enhanced survival if we compare that to normal suspension cultures. And second, we have then developed algorithms to do imaging-derived analysis of survival, of cell viability, when we add a library of small molecules, and we can then look at the responses of these leukemia cells to this library of new agents. And there we have learned that we actually see patterns of activities. So we see agents that are active throughout all different leukemias that we look at. And we see agents that only are effective in subgroups. And this is interesting from a personalized therapy point of view, because we can actually try to select samples that respond to a given treatment, at least ex vivo. We have, for instance, identified a group of T-ALL cases that responded with high sensitivity to a tyrosine kinase inhibitor, dasatinib. And this sensitivity could not be predicted by genomic analysis. And that's why we think that this functional platform is relevant, because it identifies phenotypes that we would not see if we just did a genomic workup of the samples. That's apparently a running target, so to say. So we are expanding and exchanging all the time. There is new agents that are popping up. And second, we would like to run that co-clinically to look at correlations, to what extent activity of agents that are in clinical use today correlate in the clinics with effects on our platform. We also can use this platform to look at completely novel drug response phenotypes. So we are not taking an agent and look at in what samples is it active, but we can also derive from these phenotypes information on the biology of the disease. And we find, for instance, that we can detect new targets that do not originate from a genomic lesion, but that more phenotypically drive the disease. I mentioned the dasatinib story, and another point was the SMAC mimetics that activate the kinase that we would never have thought would play a role in this disease.